Hello students. So students, in the previous class in the chapter that is life processes, we have seen the different modes of nutrition as well as we have also seen that what are the different conditions which are necessary for the plants to make their food, and for that we should know that how the plants used to prepare their food. So we know that student, the plant used to prepare their food by the process that is called as photosynthesis. So it is due to the photosynthesis they are able to synthesize their own food. Now the food which is prepared is actually a simple carbohydrate which we call as a glucose. Now this glucose is stored in the plant in the form of a complex carbohydrate, and that is called starch. Now it can be present in any part of the plant, student. It is not necessary that always the leaf is consisting of the starch, but it can be present at the any part in the form of a vitamin, in the form of a fats as well as a protein. But starch is present there. So the presence of the starch itself explains that plant has prepared the food, because starch is the food only. It means plant has prepared the food, means plant has performed the process of photosynthesis, students. Now you see here, when we were talking about the process of photosynthesis, we have seen that the condition which is necessary for the plants to perform the process of photosynthesis is, first one is the sunlight, second is the presence of carbon dioxide, third, presence of water, fourth is the presence of chlorophyll. These four are the conditions which is really very important for the plants to perform the photosynthesis. Now I am telling you that, okay, these are the conditions which is necessary. But the question arises in your mind, that is, how can we say so? How can we say that, okay, that sunlight is very important or chlorophyll is important? We can also see that the process can occur in the presence of carbon dioxide and the water. How can we prove that, okay, the sunlight is important, because if there will be no sunlight, no photosynthesis will occur. So to confirm this, students, that the process occurred by the presence of all these four different conditions, experiment has been performed, and with the help of the experiment we can prove that the process of photosynthesis requires sunlight, it requires chlorophyll, or it requires carbon dioxide. Okay, so moving on, and we will see that first experiment, in which the experiment explained that sunlight is necessary for the process of photosynthesis. Sunlight is necessary for the process of photosynthesis. For that, student, what we have to do? I told you that whenever we will perform any experiment to confirm that whether this is necessary for the photosynthesis, we will confirm it by the presence of starch. You know that the result of the photosynthesis is starch. It means if a plant is consisting of a starch, we will say that photosynthesis has taken place. If no starch is there, so we will say that no photosynthesis has taken place, because the result of the photosynthesis is the starch only, student. Now you see, how we will confirm the presence of the starch? That is also an important one, that with the help of the iodine solution. You see that whenever we will put iodine solution to starch, it will turn blue-black in color, which confirms the presence of starch. So the things that we have to keep in mind, that is presence of starch. Presence of starch confirms process of photosynthesis has taken place. Okay. Turning blue-black color after adding iodine solution in a starch, iodine solution in starch confirms its presence. You see student, this is really very important to initiate the experiment, and that is confirmation of the photosynthesis. How we will confirm the presence that photosynthesis has taken place? By seeing the result, and that is the starch. And how we will confirm the presence of a starch? By adding the iodine solution, which will turn blue-black color. Okay, so this is the two things that we should keep in mind. Now let us start the experiment. For this students, what are the different points which is included in this experiment? So the first thing that we will do, that we will take a potted plant. Potted plant, we will not going to pluck any leaf or any plant, but we will take a plant which is planted in a pot. So we will take a potted plant. Now, especially we will going to take a plant which is having a long leaf or which is having the leaves, broad leaf. That will be much better to understand. That will be help us to understand the concept very easily. But a person can take any kind of a potted plant with green leaves. Remember, with green leaves. So we will take a potted plant that consisting of a green leaves, and at first we will keep it inside in a room for 2 to 3 days, that potted plant, so that the plant will become de-starched. Now what is this? The meaning of de-starch. You see, the presence of the starch explain about the photosynthesis. So, if suppose we are going to continue with the same potted plant without de-starching, so it will going to interfere in our experiment. So, at first we will de-starch the plant. Now, how we will going to de-starch the plant? When you keep the plant for 2 to 3 days in a room or away from that of a sunlight, then what will happen? There is no sunlight, so no photosynthesis will take place. Now plant need food to survive, so they will start taking the food which they had stored in the plant in the form of starch. You know that plant used to make glucose, and this glucose is stored in the plant in the form of a starch. So when we keep the plant for 2 to 3 days, because of the absence of the photosynthesis, no new starch, no new glucose is formed, since no photosynthesis is taking place. But plant need food to survive, so they will take the stored starch present in the plant. Okay, so by keeping it for 2 to 3 days in a closed room away from the sunlight, we are able to make it de-starch. So the first point that we can write it, that for that potted plant de-starch. How? By keeping it away from sunlight. Okay, so this is the first thing that we can do. Now after 2 to 3 days, you will find that this plant is now de-starched completely, students. Okay, now what you will do? We will take that particular leaf without plugging it. We will take any of a leaf of that plant and we will cover the middle portion of the leaf with the help of an aluminium foil and press it gently so that that middle portion of the leaf covered properly. Like if suppose this is the leaf, students, so we will going to take the aluminium foil, aluminium strip and we will cover this portion, middle portion of the leaf. Okay, so at the both of the side, not from the upper side but from both of the side. So we had covered only the middle portion, leaving the rest of the tip, and still this plant is attached with the plant. Okay, so sorry, this leaf is attached to the plant. So we have taken only one leaf to for this experiment, and we will keep it the plant in the closed room to de-starch it, and then we will take out we will select any of the leaf and cover the middle portion of the leaf with the help of the aluminium foil, students. Okay, now we will cover it, and with the help of the any clip or with the help of the tape itself, anything, we will cover it in such a way that it will not going to absorb any kind of a sunlight, or sunlight will not reach at least to this portion, this middle portion of the leaf. Now, rest of the part is still exposed to the sunlight. It means when you keep it outside, it will remain exposed to the sunlight, but this middle portion should not be exposed to the sunlight. So the second point that we will going to write here, that is cover middle part of a leaf with the help of aluminium strip, aluminium foil or aluminium foil strip. Okay, so we have covered the middle portion. Now what we will do, we will take that potted plant which we had kept it away from the sunlight, again exposed to the sunlight. We will take it out from where it will get a sufficient amount of the sunlight, student. Okay, so the third step is, now we will going to keep this potted plant outside exposed to the sunlight. Exposed to the sunlight. So we will keep it exposed. We will keep the potted plant outside for 2 to 3 days, and after that, what we will do, we will going to plug that leaf in which we can paste, or we had covered with that of the aluminium foil. So we will take that particular leaf. Okay. Now after plugging that of the leaf, the student, we will keep it we will boil that particular leaf in a boiling water. Why we will do that? We will do it so that the cell membrane, it will help in loosening the cell membrane, and ultimately it will provide, or we can say that it will allow more absorption of the iodine solution to give the result. Okay, so after plugging that particular leaf, we will remove the aluminium foil, that middle portion where we had cover that portion with the aluminium foil. We will remove the aluminium foil and we will keep it in the beaker to boil it, and that beaker consisting of the water. So we will boil it with the help of the boiling process, and this will help in loosening the cell membrane, which will ultimately help in absorption or more absorption of the iodine solution. Now we have done here the fourth point, student, fourth step in which we will boil it. Now we will take it out, and now it is ready for the iodine solution. But remember, still that color of the leaf is green in color. So this green in color, if suppose in a green color, if you put the iodine solution to test that whether it consisting of a starch or not, or whether it will change the color to blue-black. So you think that while adding this iodine solution, this green color will going to interfere. Okay, so if you understand, if suppose I am writing something with the help of a black marker, if I will writing with the help of a black marker, if I am writing on a green board rather than on a white board, absolutely you will say that the green board will going to it will not gives a proper visibility or of the words, or the words will not appear clearly. But in case of a whiteboard, you can see the words clearly, you can see the sentence clearly. The same thing happened with the leaves also. When you will put the iodine solution on the green portion, so it will going to interfere to the result. So what we have to do, we have to remove this portion, this chlorophyll portion, okay, this green portion. So how we can do that? We will keep this leaf in the alcohol, okay, and we will heat it. Now we cannot heat the alcohol directly because alcohol is highly inflammable. So when you provide heat directly to that of alcohol, it will it will catches fire, and ultimately the leaf will also get destroyed along with that. So we will water bath. Okay, so how? So in which what we can do, student, we will take a beaker, and that beaker consisting of water and heat, and we will heat it by providing heat with the help of a burner, and in a test tube or in a small beaker, students, we will going to keep the alcohol, we will add alcohol, and in this one the leaf is present. So when the heat will reaches to this water, the water start boiling, and due to the heat of this water, this will help in the boiling of the alcohol, and the term that we use for it is the water bath. So what we will do, we will use alcohol to water bath, and that alcohol will help in removing the green color of the leaf. Okay, so this is the next point, and very important point. So we can write it here, and that is putting the leaf in alcohol and water bath, and alcohol and water, leaf in alcohol and water bath it. Okay, so we can write it that this will help in removing the color making it colorless leaf. You see what happened when you keep the leaf in that of the alcohol, student, and start boiling, start giving water bath to it by heating the water, and ultimately the alcohol will be boiled, you will find that before that, when you had put that leaf in the alcohol, at that time it was colorless solution. But as the alcohol start boiling, the color of the alcohol changes to green in color. And the reason, you have understood that the color of the leaf starts coming out and making the solution green in color, and the leaf itself become colorless. Okay, so we had given water bath to that of the leaf by keeping it in the alcohol. Now what we will do, student, we will take out the leaf from the alcohol. Now it is a colorless leaf, and again keeping it in a boiling water in order to remove the extra alcohol and making it more loose. Okay, now we have taken out that particular leaf also from the water, and now this time it is ready for the iodine test. So what we will do here, so we will take that particular leaf, we will take that particular leaf, and now when you will add the iodine solution, what you will find that the portion which was covered by the aluminium strip, it does not gives blue black color. But the portion which was exposed to the sunlight, which was not covered with the aluminium foil, turns blue black color on adding iodine solution to it. Okay, so basically we have seen here one thing, that when you added the iodine solution drop by drop to all the portion of the leaf, you will find that the portion which was covered with the aluminium strip, that does not turns blue black color, but the portion which was not covered, that turns blue black color. So you see, student, what we had done. At first we have de-starched the leaf by removing all the starch present in it and cover it with that of the aluminium strip, and because of the covered portion, it did not receive the sunlight when we had kept this potted plant exposed to sunlight. Okay, and since it does not receive the sufficient amount of sunlight, so there is no process of photosynthesis has taken place at this portion. Okay, why? These two portions, they had received a sufficient amount of sunlight. So because of the presence of sunlight, they are able to synthesize food, they are able to show the process of photosynthesis, and you know that the result of the photosynthesis is glucose, which is stored in the form of a starch, and it means that these two portions consisting of a starch in it, and we had confirmed this with the help of the iodine solution. So when we had added the iodine solution with the help of a dropper at both the places of the leaf, it turns blue black color. So which proves that these two portions which was exposed to sunlight has performed the photosynthesis, while the portion which was not exposed has not performed the process of photosynthesis. So it does not consisting of a starch. So this whole experiment has explained here one thing, that sunlight presence is really important. It is a condition which is necessary for the process of photosynthesis, because if no sunlight will be there, there will be no formation of a starch, and no formation of a starch confirms that there is no process of photosynthesis has taken place. So to carry the process of photosynthesis, sunlight plays a very important role. It is that condition which is mandatory, which is necessary for the photosynthesis to be occur. Okay, so this is the one experiment. Now, Okay, so this is the first experiment. Now moving on, and we will going to see the other condition which is necessary for the process of photosynthesis, and how can we confirm that that is the important condition for the photosynthesis. So moving on, and we will see the next condition. So the next one is the chlorophyll. Chlorophyll is necessary for the photosynthesis. How can we say that chlorophyll is important? So for this experiment, at first we will take a leaf which is partially white and partially green. And such kind of a leaf is also called as variegated leaf. Variegated, it means variety of a color are there. So basically we will choose that leaf which is partially white from outside and in middle it is green in color. Okay, so that will also help in confirming that the white color does not consisting of a chlorophyll, but the green color consisting of the chlorophyll. We are knowing this concept, that is green color consisting of a chlorophyll. It is green because of the presence of the green pigment, that is chlorophyll. If it is white, so it does not consisting of a chlorophyll. Now at first we will going to take a leaf which is partially white and partially green. Okay, so we'll take a potted which is partially white and partially green. Now we will keep this again we will restart the plant. So we will keep this potted plant inside away from the sunlight. Why we used to do that, student? We used to do in order to remove all the old starch which is present in it, and by starting or we will going to start an experiment with a fresh leaf, okay, which does not consisting of a starch. So the second step will be de-starching the leaves by keeping it inside, by keeping it away from that of the sunlight. Okay, so second point is de-starching. De-starch it. I am not writing the whole sentence. I hope you have understood how we can de-starch it. Now again we will keep it by de-starching. Now again we will keep that potted plant outside exposed to the sunlight, so that the further photosynthesis or a new process of a photosynthesis can take place in the plant. So what we have done, we have taken a plant which is partially, whose leaves are partially white and partially green. Then we will keep that plant inside away from the sunlight, so that the process of so that all the starch which is present in it will be used up, and again we will keep it outside exposed to the sunlight, so that the process of photosynthesis can take place again. Now what we will do, after 3 to 4 days we will pluck the leaf, that variegated leaf. That variegated, it means that leaf which consisting of a white and green color. Now what we will do, student, we will do the iodine test by again doing the same process. It means by boiling it in the water so that the cell membrane will be loose, or after that we will keep it in alcohol and we will do the water bath by keeping the leaf in the alcohol and boiling the water. And because of that, what happened, the color of the leaf will be released by the leaf, whether it will be the green color which is present in will be removed. Now we will again take out that particular leaf, boil it again so that the cell membrane will become loose, and then it is ready for the iodine test. Now when you add the iodine solution on that variegated leaves, you will find that the portion which is white color, which was white color, before that which was white color, it will not going to change. There will be no such change occur. It will not going to turn blue-black in color. But the middle portion which was green in color will turn blue-black in color. Okay. So this will help in proving that the outer portion does not consisting of a chlorophyll, so it does not changes blue-black in color. It means it does not consisting of a starch. While the middle portion, it turns blue-black color. It means it consisting of a starch, and it performs the process of photosynthesis. Okay.